So practical, we will be focusing on water microbiology. So here, if you are given one sample of water and you are asked to enumerate the both culture dependent bacteria and culture independent bacterial abundance in the water sample. So for culture dependent approaches, we'll be doing spread plating and membrane filtration technique, whereas for culture independent enumeration, we'll be carrying out direct counting using the DAPI stain. Okay, so you can see the techniques for spread plating, membrane filtration, and direct counting through this YouTube videos. Okay, so in the practical schedule, it is stated that the acridine orange AO is used as the fluorochrome, but nowadays we use DAPI more because the colors are brighter and clearer. So for the procedure for DAPI staining is usually about 100 microliter of stain in 2 ml of sample. You gently mix it and then you leave it to incubate for about 7 minutes before slide preparation. So all these steps are available in the YouTube video, so please look at them. Then this formula here is for the calculation of cells per mil for the direct counting via DAPI. So the principle here is this is the area of the microscopic field, whereas this is the area of the filterable area. Okay, so after you've done direct counting, you will be able to measure the filterable area here, and then you can, from the microscope field, you can measure or determine the area of the microscope field. So the ratio of these two multiplied with the sum of counted cells inside the microscope field will give you the cells per mil of the water sample. Okay, so because this is the area of the filterable area and this is the microscope field, so if you are using a different microscope, or if you're using different lenses, or if you're using a different kind of filtration set, you have to remeasure this the diameter of these areas and calculate the area again. Okay, so you have to measure the diameter and then calculate the area again. So these are the sample results. For spread plating, after carrying out zero dilution at 10 power 0 minus 1 minus 2, you obtain CFU counts like this. Calculate the CFU per mil here using the standard guidelines. Then for MFT also, you have your counts here. Calculate the CFU per mil. Please take note that you are filtering 0.1 ml of 10 minus 1 dilution. So 0.1 ml is a very small volume, so what we usually do is we'll add about 5 or 10 ml of sterile distilled water first before adding the sample inside and then filtering the whole volume. This will ensure better distribution of your CFU. And finally, for the DAPI staining, we are using the direct count method, and these are the cells per field that were observed. Okay, so from here you can also estimate the cells per mil. So these are microscope fields, micro photographs of Dumpy stain samples. So the blue dots here are the bacteria. Okay, similar here. The blue small dots here are the bacteria. So if you have a very large patch like this is not bacteria because it is too small. It is too big to be a bacteria. Bacteria. This is at uh this is about 1,000 times magnification. The bacteria should be this size. So sometimes inside your own sample, inside the same sample, if you change the filter set of the epifluorescence microscope, you can look for other colors. So here we can see the autofluorescence of the pico cyanobacteria. Cyanobacteria. So these are most probably Shinicococcus. You can see cells here. Okay. So these are the questions that I hope you will take time to answer. What does Dumpy stain? So what are the colors observed? And what is the purpose of a backing filter? What is the percentage of the culturable bacteria over total? And given the different methods available, quantifying bacteria in water samples, discuss the advantage and disadvantage of each method. Okay, so if you have a data set like this, you are using 3 methods here. So if you want to compare them, what is the best statistical approach to use to be able to compare them? Okay, please try uh, carrying out some form of statistical analysis to see whether you are able to do this, compare these 3 methods.